Phototransduction in Light by Hyla Torres. In our unit of sensory organs in the peripheral system, we will today discuss visual sensory receptors and processes in the human body. At this point, we know that vision is perceiving objects within an environment by means of light that is reflected or emitted to us in some manner. We have noted that light is a type of visible electromagnetic radiation. To better understand the topic today, we must first be able to properly identify and define the basic anatomy of the eye. As a brief review, we will start frontally with the cornea. The cornea is in the anterior region of the sclera that emits light to the eye. Between the posterior chamber, anterior chamber, and iris, a liquid substance called aqueous humor flows. The lens is made of tightly compressed fibers called lens fibers that is suspended in place via the suspensory ligament. It is crucial to understand these anatomical parts of the eye to understand how light enters the eye to begin the phototransductory process. Prior to going through the pathway of how phototransduction occurs in human physiology, let's first define it. Phototransduction is the mechanics of converting light stimuli into electrical activity by the rods and cones of the eye. This means the body will take the light that someone is exposed to. It is then converted into various electrical signals. The cycle in which the light processes is called Wald's visual cycle, named after the founder, George Wald. He even won the Nobel Prize in 1967 for his discovery. This is a relatively new discovery, and it's important to keep in mind even though we think seeing things is easy, it's actually quite a process. So let's kick things off by discussing the main components of how phototransduction occurs by naming the key players of the process: rods and cones. Rods and cones are the photoreceptors found in the human body system retina. Remember, photoreceptors are a sensory organ that respond to light intake. It is a highly specialized type of optical nerve. Rods specifically assist in vision in low light settings. They do not aid in anything to do with visually seeing color and have a low ability in spatial recognition. Rods are typically found in the outside perimeter of the retina, which is in the back of the eye towards the optic nerve. Rods will absorb or take in light from an outside source and convert it into a neural impulse. There are more rods in a retina than there are cones. Cones, on the other hand, are in fact capable of both color vision and spatial recognition. They are also optical nerves and photoreceptors. There are various types of cones with various functions. Cones are typically found in the middle center part of the retina, near the center named the fovea. It's important to note that photoreceptors do not themselves spur on action potentials. However, they can trigger other cells that can fire action potentials. This will be discussed here shortly. Rhodopsin is a membrane protein of the retina and is the visual pigment in the rods. Cones have a similar protein, but it's named photopsin. Each disc within the rod is densely conjoined with these globular proteins. To maximize light absorption, the membranes work to hold them in place. The retina is unique to other sensory systems, as photoreceptors do not demonstrate action potentials. Most sensory systems are the opposite. Light activation can lead to changes in the membrane potentials and will ultimately lead to a change in release of postsynaptic neurons. Specifically, within the retina, graded potentials occur. Here, action potentials are not mandated to convey data because it is a small distance. To put all the pieces together here, imagine the light entering the eye It'll initially hit the lens. It then goes on to the retina, which is towards the back of the eye as mentioned. Inside the retina is where millions of these photoreceptors, the rods and cones, are found. When the light hits the rhodopsin inside of the rod, it will then begin the phototransduction process. And this is when the particular rods affected by the incoming light will eventually trigger the functional cells to fire an action potential. It sends electrical signals that the brain receives. Bipolar and ganglion neurons are a part of this pathway. Bipolar neurons have synapses on one end and are connected to a ganglion neuron on the other end, which forms the start of the optic nerve. Ganglion cells are the largest neurons found within a retina. In these ganglion cells is where the action potential is fired. These cells usually receive an input from multiple bipolar cells. When the action potential is carried on from the ganglion cells, it goes through the optic nerve and sends impulses to the brain, specifically the visual cortex. In this photo, you can see the cones and the rods, the epithelial cells, To see how research has progressed in studying the phototransduction process, a recent study displayed how the route of light signaling travel takes place in the rods of a mouse in vivo. More specifically, the study conducted was designed with a rate equation model to see how rhodopsin may impede on diffusion of molecules on the disc membrane. The manner begins when photoreceptors are activated via membranous disc. From there, negative feedback via activation and suppression waterfalls forms proteins. From proteins, molecules and ions that are found within the cell of the rod. These results of the study are in the in vivo circumstances displayed how this type of molecular crowding caused an increased amount of protein folding, stabilization of intracellular architecture, and more. Ultimately, it leads to highly bound biochemical processes like signaling proteins pattern formations. A theoretical model of the phototransduction process was created in mutant mouse rod cells so that the researchers could discover how the mechanisms worked in various light intensities. Models have been studied where secondary messengers have been observed within the cell cytoplasm. The purpose was to identify features of the phenomena. However, the trials have yet to have an official conclusion drawn. In this image, we see the microscopic world of the phototransduction process. Here, we see a rod portrayed. Please note that the same idea can apply to cones. Cyclic GMP-gated channels will be accountable for the changes made from light input electrical signals. On the contrary, when it is dark, the cyclic GMP levels increase. cGMP is able to then bind to Na+ permeable channels within that very same membrane and will encourage sodium and other various cations to enter the channels. It will then begin to depolarize the cell. When the darkness ceases, the eye is exposed to light. cGMP will decrease and close the channels.